Professor Tony Watts, it's a pleasure to have you here. You work with cell membranes. They separate the interior of cells from the outside environment and are the first point of contact between the cell and the world. What happens at this interface?
Yes, that's right. Yes, I've been working on cell membranes all my research career. And in the early days, membranes were thought to be just lipids and proteins, but the chemistry and diversity of these molecules was really not well known at all, or even characterized. And indeed, nobody knew whether membranes had any functional influence on the cell and how it functions apart from protection from the outside environment. But early in my career, in about 1973, a new model for cell membranes was suggested, and that was called the so-called fluid mosaic model for membranes. And testing this notion that membranes are highly dynamic is still being investigated. It's still a major challenge, so still today. For example, the motions as fast as femtoseconds can occur in certain light capture proteins, such as in your eye, but then very slow motions, such as the turnover of your red blood cells, takes about 120 days, are all encompassed within biological membranes. And the structure of a membrane is such that you have two layers of lipids with proteins either on top or embedded within this bilayer. So on the outside of the bilayer are so-called polar groups that like to interact with water. But on the inside, you've got fatty chains, which are much like oil or grease. So it's very different chemically. And it is amazingly robust and stable structure. And it's this dual chemistry of lipids, which is absolutely fundamental to their integrity. So imagine a cell about the size of a living room in a house. The membrane, to put it to scale, is about 4 nanometers in diameter, but in that scale, it would be like a party balloon blown up inside the room. That's the kind of scale that we're talking about here. So it is an amazing structure, and it's staggering to imagine. So indeed, interesting, some suggestions about the beginning of life on Earth. It's proposed that lipids were made in the atmosphere, and some of these self-assembled, maybe on rocks, into bilayers to make a compartment, much like a very primitive cell, and then inside the cell life developed. And much has been written about that process. So the chemistry is really quite fascinating.
Yes.
Sorry, nowadays we know there are over 40,000 different types of lipids. This is a major area of research called lipidomics, usually using mass spectrometry. And now we know membranes are really very complex. They've got glycolipids containing sugars, proteins, scaffold proteins, and so on. And they really do influence the whole of the cell in its many functions, from changing composition and multiple interactions. It's a very fascinating area of research.
Yes, you investigate lipids, proteins, and their interactions. Could you talk a little bit more about lipid properties?
Yes. So the lipids themselves, I say they have this water-loving groups on the outside, polar groups, and the fatty parts in the middle. And that gives them a very special property. They can self-associate, they can aggregate, they can form little particles within the membrane. And one thing that is important from the physics point of view is that the outside water component has a dielectric of about 80, but the inside lipid component has a dielectric of about 2. Now, for those who are physicists, they will realize that that's a huge gradient of dielectric. And then when you remember that the proton motive force, the voltage across most living membranes, they are electrical insulators, is about 300 millivolts, for example. That gives you a voltage gradient phenomenally of 10,000 volts per centimeter. And that's a phenomenal charge gradient across, potential gradient across the membrane. No man-made insulator or capacitor can deal with that kind of voltage without breaking down. So nature has solved this problem of maintaining electrical integrity across the membrane and protecting it from the outer environment. So it has some very fascinating physical and physical-chemical properties as well. So these are the lipids themselves, and physicists explore lipids in phase diagrams. Chemists make lots of different kinds of lipids, and biologists investigate how these lipids interact with the proteins to determine the cellular function.
Concerning proteins, you've been studying the family of G protein coupled receptors and photoreceptors.
Sure.
Why is it important to know the mechanisms of action of these receptors?
Well, as an observation, the Nobel Prize in Medicine awarded just 2 days ago, and this joins previous Nobel Prize when a prize is awarded just for the research of G protein coupled receptors specifically. That's how important they are. So indeed, these proteins, we call them GPCRs, G protein coupled receptors, constitute actually the largest family of proteins in high life forms. That might sound a lot, but there are only about 800 or so that we know about which have functions, but still they have very significant functions. For example, they respond to signals such as hormones. They respond to photons of light in photoreceptors, as you mentioned, and they turn these signals into a signal for the organism. It tells the organism to do something. This could, for example, be metabolic control, important in dietary control. It could be in sensory perception, for example, heat and pain, as in the most recent Nobel Prize work, or even taste, hearing, and so on. And then even to gene function, they can change the gene function of a cell. So it's a very, very complex set of interactions, which we don't know almost nothing about still. Very few structures of these proteins are known. It's incredibly complicated. It'll keep scientists going for a long time to come. But interestingly, one signal at these GPCRs can initiate up to 1 million biochemical events. This is actually happening at the back of your eye at this moment. One photon can initiate 1 million electrical events to the optic nerve to the brain. So it does that in about a microsecond, and the initial events occur actually in about femtoseconds. So no man-made amplifier is as good as a GPCR. It's a 60 decibels amplification in a microsecond. There's nothing that man can make. So man can learn a lot from biology and from just these kind of proteins, but many other proteins as well. And I've been working on those photoreceptor proteins, as you say, for a long time, both the ones in bacteria and the ones in your eye. And in fact, these light receptor proteins are some of the most abundant on the face of the earth. So it's a very fascinating area of study. They're also used in optogenetics, for example, as markers in the brain and brain cells to look for neural networks. And that's a big area of study, optogenetics, and these photoreceptor proteins help us visualize neural signals. So GPCRs and many membrane proteins, a lot to teach us. And we're still very much in the so-called hunter and gatherer phase of biophysics, finding out information and putting it all together and finding out how the structures work. So they are a fascinating area of proteins, I say.
Could you talk a little bit about lipid nanoparticles technologies that you use to study these receptors?
Sure. Yeah, so nanoparticles are everywhere in nature. Viruses and bacteria are very good examples, actually. And recently, we've been able to, as scientists, and us included, have been able to make nanoparticles out of lipids. So these look like a little bit of a natural membrane, and that's really quite a big advance. In our own work, we've used those lipid nanoparticles to deliver effectively chemotherapy drugs to several organs of an animal in a viable way, for example. And clearly this approach has the potential to deliver drugs in a range of areas. In fact, the recent vaccine development by Pfizer and Moderna, driven by the COVID pandemic, of course, has been enhanced significantly by the use of lipid particles to put the mRNA into, to be able to deliver that in a vaccine. Fantastic example of translational biophysics. So in other studies that we've done, in fact, we've used these nanoparticles to look at the lipids in a worm, C. elegans, which is a very popular model in biology for genetic studies. And by looking at two different worms, wild type and one that's resistant to bacteria, we've used the characterization of the lipids to show that the mutant, which is resistant to bacterial infection, has a different lipid component on its outside cuticle from the wild type. So we can understand that in chemical terms now. So a worm can develop the bacterial resistance just from changing its lipid component. So nanoparticles are really very interesting, very important, and they also can help us study membrane proteins in nuclear magnetic resonance, crystallography, mass spectrometry. We've used it for all of these methods. So we're exploiting nature to give us more information and to help us.
Why is it a challenge to understand how biological membranes work?
Yeah, well, as I've said before, there's a huge challenge in understanding how all of the different components of a cell interact not only with each other, but also with the outside environment. For example, at the moment with COVID, how a virus attacks a membrane, takes the membrane apart, goes inside the cell, and then replicates, and then, of course, it produces more virus particles. So all of that is really still quite difficult to understand in many ways. There are very many complex interactions going on there. And so one of the major problems at the moment is to couple any biophysical or structural approach with the function of the cell or with the function of what's happening, the outcome. So we might often know a lot about the structure of a protein, but how that in a membrane with the lipids and everything else derives a final function, there are many, many steps, and putting those together is quite difficult. And it's often the functional studies are phenomenological without the structure, and many of the structural studies are without the function. So there's a big disconnect, in my view, between these two things. I see that in computational studies, for example. So there's a lot of computation goes on in the world at the moment. Computational studies are maybe a third or maybe even up to a half of all kind of biophysics and scientific research, which is fantastic to see and to do. But many of those for membranes don't say how the computational side connects with the function. And that's really what we want to know. And I think that's where the next major challenge will be. And unfortunately, the functional part is often ignored, which is a shame. Okay.
You already mentioned some of the methods you use. Could you briefly describe the methods?
Yeah. So, yeah, I've used a range of methods, a very wide range of biophysical methods for my research over the years. And as I've said before, my major driver has been to explore the dynamics in membranes and then link those to function. And so many of the methods need to be adapted for use in membranes. And that's one of the things that we've been involved with. Biophysics often is a development of methods and make them so that they can be used on membranes in all their complexity. And it's spectroscopy is one of the areas in which I've been working a lot. For example, nuclear magnetic resonance and electron spin resonance, each of which have their own intrinsic timescales of observation. So NMR is very relatively slow motions in the millisecond time range. The electron spin resonance is very fast motions, typically in the nanosecond range. So trying to couple those has been one of the things that we've managed to do. And some of those methods require chemical labeling. So we need to do chemistry because we need to exploit the magnetically active species we want to observe, for example, deuterium 2H or carbon-13, and also the methods themselves need to be refined in some way. Membranes are very large macromolecular complexes, and they're really tough to handle, in particular for some methods. And recently I've used microscopy and crystallography, and a recent exciting project has been to use X-ray free electron lasers to gain information about motions within proteins. For example, the photoreceptor proteins shine a laser at it to excite it in femtoseconds to milliseconds, and then immediately after we use a high power laser to get a structure, so we can see structures at different time periods during the function of the protein. That kind of work is available in places, only 1 or 2 places in the world, like Sackler in Japan or LCLS at Stanford in California. And it really is an exciting move forward. So I try to keep up with lots of different methods. I use a method to answer a question. I don't define a method just for the method's sake, which is some areas of science. I ask the question, then I find the method to answer the question I'm asking.
Okay. Now I would like to change the topic a little bit. How important is it for you to participate in the 20th IUPAB Congress in Brazil, despite the meeting being virtual?
Sure. Well, it's not the first meeting I'm at virtually, but it's the first IUPAB Congress I've been to virtually, of course. But the first meeting I went to was in 1978 in Kyoto. And interestingly, almost 50 years later, the next IUPAB Congress will also be in Kyoto in 2024. So the 6th Congress in 1978, and I think I've been to the vast majority of the IUPAB Congresses. They're usually every 3 years as a scientist. But now I need to be there because I have a role to fulfill within IUPAB. And attending congresses are important. IUPAB has a special flavor because it's international, and it has an intention of bringing people together who normally would not have access to the big congresses in America or Europe, for example. So they do go around the world, and it's an international flavor. And in particular, people from less well-resourced countries can come together and meet with scientists and network and work together. And for younger scientists in particular, to initiate networks and find out labs where they might like to work is very important. So in a physical sense, for a meeting, that's one of the big outcomes. In a virtual sense, you find out about the latest science. Of course, you have a chance to ask questions and posters and so on. So it's very important. This international flavor is really quite unique. And I like IUPAB congresses. They have a good flavor to them.
Well, you were elected president of the IUPAB. Congratulations. I think this is the role you said you have. I would like to know what do you expect to achieve during your presidency?
Thank you for the congratulations. Yes, so I've been involved with biophysical societies for many, many years now, both in Europe and also in the UK, of course, which has a strong biophysics community. So I'm very honored indeed to have been elected the president of the International Union of Pure and Applied Biophysics. And that includes about 60 biophysical science societies that are signed up as adherent member bodies to IUPAB at the moment. But one of the first things I'm trying to do is to increase that number. And already I've made contact with groups of biophysicists in Asia, Central America, for example, Africa. And these are relatively small groups of scientists who don't have a society, but they might get together and form a society in their own countries and give them an idea about collaborating with each other, having collaborative meetings and so on. And they probably would like to get involved with larger organizations to get into contact with scientists in a broader context across the world. Africa is a very important continent in which to increase scientific activity and awareness. And secondly, like many scientific unions, and there are unions for chemistry, unions for physics and biology, so on. They provide a lot of benefits to their members, not least in organizing congresses like this, especially in the world where scientists, as I say, might find it difficult to travel. But I think that the more kind of well-resourced countries are happy to see the different scientific unions helping countries that are less well off than they are. Other benefits they bring, of course, are travel fellowships for younger scientists to do exchanges between laboratories in different countries, and I hope to be able to be involved with that. Setting up workshops in specialist training and big facilities that they can go to around the world. So I plan to increase those outreach activities when we can take part in those again as part of my brief. And lastly, although I'm sure it'll be a busy term of office, since biophysics is often referred to as a bridging science between physics and biology. Nowadays, it encompasses mathematical modeling, chemistry, and genetics. And another aim is to try and increase the interaction with scientists in the more traditional areas, in particular young ones, because it's the young ones who have open minds and can be ambitious and have ambitions to kind of use their experience into new areas. So this, these are our future in terms of science. And so they need to be exposed to biophysics, which is often not taught. It's a relatively small area of science. It's not taught in many places in the world. And so just to raise the profile of biophysics. And the council for IUPAB is almost all new, and have a great group of council members, executive committee members to work with in all of this activity of our IUPAB. And I'm looking forward very much to working with them. Some of them are long-standing friends and colleagues, and others are new people that I've not met before. So I'm looking forward to my term of office as IUPAB president, which actually formally starts in 3 years' time. For the next 3 years, I'm president-elect. So I will be working up to being full-time president. So yes, thank you.
In talking about young scientists, what advice would you give to a young scientist?
Yeah, I remember back to those days, and it can seem tough. Go to work in a lab, the best lab you can. Travel if you can before you get into the spiral and the seamless worries of finding a tenured position and getting a permanent job and settling down. So use this time as a young scientist to take advantage of, for example, travel fellowships from IUPAB, visit new labs, go and meet people of your own age, but also senior people as well. Personally, I worked in Germany, the US, I worked in Paris, I had some limited time in Australia, and every time I visited these places, the experience was not only very good, but actually led to an enhancement of what I was working on at the time. It actually increased my scientific activity and knowledge a lot. It makes a big difference. So it's been a real privilege actually over my life to be paid for what I am interested in doing. Somebody else pays me for doing what makes me very interested. And everyone in research is adding to worldwide knowledge base, and it will have implications for future development. So I think most of all for young scientists, enjoy yourself.
Okay, thank you very much for joining us, Professor Tony.
It's been a pleasure. Thank you for interviewing me.